Good morning, everyone. Thank you so much for attending Workshop 11, Topical Advancements in CFTR Biology. I have the pleasure of co-chairing this session along with Dr. Carlos Farinha from the Faculty of Sciences, University of Lisbon. And so, as you all are aware, we do have to share this quick CFF disclaimer slide before we get into the science. And then the disclosures for myself and Carlos are listed here. Okay. Given the pertinence of CFTR-directed therapies, especially those that are nucleotide-based, small molecule-based genetic therapies, it's really important that we do not lose sight of making sure that we keep a keen focus on basic features of CFTR biology, the processing, synthesis, function, and so on. So we're really excited today to be able to cover a few different stories that are rooted in this theme. We are going to particularly emphasize CFTR expression, stability, and function with the first few talks. And then we'll round out the session with discussing the relevance of those findings and others with the genetic-based and other therapies being developed for CF. All right, so beginning, we will hear from Jing'an Chen, who will be discussing non-viral delivery of chemically modified suppressor tRNAs. Next will be Suhee Chang, discussing nascent chain misfolding and how that slows translation and stimulates co-translational mRNA decay. Followed by Jonica Brown, who will present the role of cholesterol and its effects on CFTR single-channel characteristics. And then the last two presentations will be from Norman Dallaire and as well as Deborah Baines. Norman's discussion will be about the liabilities of codon optimization, and then Deborah's talk will focus on airway targets for CF gene therapies. And I'd just like to go ahead and note right now that Deborah had a last-minute circumstance in which she could not travel to be here from the UK, but she did pre-record her talk for us, and so we will listen to that as the last presentation. All right, so with that, I would like to invite our first speaker, Jing'an Chen.
Okay. Good morning, ladies and gentlemen. I want to thank the chairs for having me here. My name is Charles. I'm a fourth-year PhD student in Dr. Bowen Lee's lab at the University of Toronto. Today, I'll be talking about our work on non-viral delivery of chemically modified tRNA rescues nonsense mutations in cystic fibrosis. There are no relationships to disclose related to this presentation. So cystic fibrosis is a genetic disease caused by the CFTR gene mutations. And among all the known CFTR mutations, nonsense mutation accounts for around 10% of the whole CF populations. However, the current CFTR modulators are not effective for those 10% CF patients suffering nonsense mutations, leaving there's no effective treatment for this population. So as shown here, that nonsense mutation usually caused by a single nucleotide change that causes a premature termination codon over here, which terminate the full-length CFTR mRNA translation that causes truncated CFTR protein. However, suppressor tRNA is a novel type of gene therapy that could potentially restore protein translation across nonsense mutations. As we can see here, suppressor tRNAs with their anticodon engineered to base pair with the PTCs, which can read through the PTC to ensure the full-length CFTR mRNA translation so we can get the full-length and functional CFTR protein. However, there are still challenges of translating this novel therapy for CF treatment. The first one is how to make the suppressor tRNA with low immunogenicity but high read-through efficiency. Second is how to deliver the suppressor tRNA to lung epithelia efficiently in vivo. So first, let's take a look at the natural tRNAs. So those tRNAs are heavily modified in nature, and those modifications are critical for the tRNA structural stability and their interaction with other factors such as the ribosome, elongation factors during the translation process. Therefore, inspired by the natural modifications of tRNA, we asked that could we introduce site-specific chemical modifications into suppressor tRNAs for enhanced read-through and reduced immunogenicity? So we first introduced multiple chemical modifications into a glycine-based suppressor tRNA, and we test all those variants in a human bronchial epithelia reporter cell line. And we find that among those chemical modifications, 3 of them showed enhanced read-throughability, as you can see here, TGM1, 2, and 6, compared to the unmodified sequence. And among these 3 sequences, our lead candidate is TGM2, shown here, where it showed around 16-fold change in terms of the protein restoration from nonsense mutations. We also test the immunogenicity of those chemically modified suppressor tRNAs in human monocytes, or THP1 cells. And we showed that all those chemically modified variants showed reduced immunogenicity compared to the unmodified sequence to ensure their safety as well as their potential suitability for multiple dosing. As I mentioned before, so the chemical modification that we investigated was based on a glycine suppressor tRNA, and that's because G542X was one of the most common nonsense mutation in CF. However, there are also other type of nonsense mutation in CF, such as R553X or W1282X. Therefore, we applied the lead chemical modification that we identified into arginine and tryptophan suppressor tRNA as well. And we find that the lead chemical modification consistently enhanced PTC read-throughability of various suppressor tRNA, meaning that this method could be generalized. Next, we also proved that those chemical modifications could prolong the suppressor tRNA effectiveness persistence, shown both in vitro and in vivo. We can see that the chemically modified suppressor tRNA effectiveness can last for more than 20 days, which significantly outperform the unmodified sequences. So next, I'll talk about the delivery challenge. So ever since the success of mRNA vaccine for COVID-19, lipid nanoparticles have been identified as the optimum delivery vehicle for RNA delivery in the clinic. Therefore, lipid nanoparticle or LNPs should be suitable for pulmonary tRNA delivery as well. So for LNPs, they consist of 4 components. First one is ionizable lipid, as well as cholesterol, helper lipid, and PEG lipid. Among those components, ionizable lipid is the most important component that can determine the transfection efficiency as well as the cell targeting of the lipid nanoparticles. Therefore, we aim to combinatorially design a novel ionizable lipid for pulmonary tRNA delivery. To achieve this, we utilize combinatory chemistry, which is a Woogie-based 4CR reaction here, that we combine 10 different head groups, 5 different linkers, 5 different tau A's, and 4 different tau B's to simultaneously synthesize 1,000 chemistry distinct ionizable lipid for tRNA delivery. And those 1,000 ionizable lipid are formulated with the rest of the components as well as the suppressor tRNA into lipid nanoparticles. And we first screen those 1,000 different LNPs in human bronchial epithelia reporter cell lines to identify the lead candidates. And then we perform formulation optimization of those lead candidates or lead ionizable lipid in air-liquid interface culture or ALI culture. For these ALI culture, we apply artificial mucus in the insert to mimic the thick and sticky mucus that was found in CF airways, so that we're making sure that our lipid nanoparticle can not only transfect the epithelial cells, but also penetrate this mucus layer in the CF airway. After these procedures, that leads us to the identification of a lead LNP, where we call it TTP3, that enables efficient pulmonary suppressor tRNA delivery. To validate this, we utilize a transgenic reporter mouse model called LumiA mouse, where it contains a luciferase gene that has a PTC in it. So if we deliver the suppressor tRNA, that will read through the full length of the luciferase to give us luminescent signal to see the PTC read-throughability. And after intrathecal injection of the LNP encapsulated suppressor tRNA, we find that our lead LNP achieved substantial pulmonary suppressor tRNA delivery that outperformed the commercial lipid nanoparticle, which is MC3 here in this case. So now that we know that our LNP could efficiently transfect the lung with suppressor tRNA, we also want to know what cell types specifically can be transfected. To achieve this, we incorporate a fluorogenic aptamer that enables tRNA visualization for biodistribution study. So similar as before, we intratracheally deliver the LNP encapsulated tRNA aptamer. Using flow cytometry, we confirm that our lead LNP has a dominant epithelial cell transfection instead of endothelial or immune cell transfection, which is beneficial for CF treatment. However, for the commercial LNP, it actually have a dominant endothelial cell or immune cell transfection instead of epithelial transfection. And among all the lung epithelial cells, there are some key lung epithelial cell types that are central to CF pathology, such as ciliated cells, club cells, and basal cells. And using both flow cytometry and immunofluorescence staining, we confirmed that our LNP could achieve high transfection efficiency in those three cell types as well, including around 22% in ciliated cells, around 90% in club cells, and around 32% in basal cells. So now that we have the delivery vehicle as well as the effective gene therapy, which is the suppressor tRNA in this case, we next start proving the therapeutic efficacy of this system. First, we proved that the LNP suppressor tRNA system can restore CFTR protein expression in vitro. In this case, we used the 16 HBE cells carrying CFTR nonsense mutations. And confirmed by Western blot, we do see that the suppressor tRNA could achieve CFTR protein rescue in both G542X and R1162X nonsense mutation cells. And those CFTR protein expression are restored, and we also proved that the suppressor tRNA system could restore the CFTR function in those cells as well. Confirmed by the Flipper assay, we do see the CFTR ion channel activity is being restored in the treated cells. Moving from in vitro to in vivo, we utilize this CFTR R553X mouse model, and we intrathecally inject the LNP suppressor tRNA system into the mice on day 0, day 2, day 4, with 3 dosages in total. And at day 6, we do find that in the treated mice, there is a significant higher level of CFTR mRNA expression level. And to prove the functionality improvement of CFTR after treatment, we also extract the intestinal organoids from those CF mouse model. And we can show here that after the suppressor tRNA treatment, using the phosphocholine-induced swelling assay, those treated intestinal organoids showed much higher ion channel activity compared to the untreated intestinal organoids. Last but not the least, we also extract patient-derived intestinal organoids. This CF patient contains 2 nonsense mutations simultaneously, including S466X and R553X nonsense mutations. So as shown here, we can see that this patient-derived organoids that contains 2 nonsense mutations are not responsive to either G418 treatment or the modulator treatment, which is Trikafta in this case. However, we do see a increasing trend of CFTR ion channel activity improvement after the LNP suppressor TRNA system treatment. And when we combine the LNP suppressor TRNA system with Trikafta, the modulator, together, we do see there's a significant improvement of CFTR function in those patient-derived intestinal organoids. So to summarize, we first find that chemical modifications could enhance the therapeutic efficacy of suppressor tRNAs, where we find there's improved PTC resorability, safety, and persistence. We then utilize high throughput screening to identify a lead lipid nanoparticle for pulmonary tRNA delivery, where they achieved dominant lung epithelial transfection, and also, as well as a higher transfection efficiency in key lung cells central to CF pathology, such as basal cell or club cells. Last but not the least, we proved the therapeutic efficacy of this LNP suppressor tRNA system in terms of recovering the CFTR protein expression and function in various CF models, including the HBE cells, CF mouse model, and patient-derived organoids. Last, I want to thank my supervisor, Dr. Bowen Lee, as well as all the collaborators and funding agencies that contribute to this work. Due to the time limit, there are some detail that's not covered in the presentation, but I'm happy to discuss any issues and answer any questions. Thank you.
Thank you very much for your talk. So this is now open to questions. Greg.
Greg Hurlbut, Siona. That was a great talk. I'm curious, what effect do your suppressor tRNAs have on naturally occurring stop codons?
Oh, yeah. Thank you very much. So that is a very common question for the suppressor tRNA field, that whether we change the anticodon of the suppressor tRNA, whether they could race through the normal stop codon, causing the longer length of normal proteins. The short answer is that it won't, because we perform ribosome profiling to confirm that the normal stop codon is not being raced through by the suppressor tRNA. But the specific mechanism for that is not clear yet to the field. But we do believe that the mRNA translation process, because of the structure change of the mRNA, such as the 3' UTR being folded to the 5' UTR, that cause a structure change of the mRNA. So when the suppressor tRNA, even with the suppressor tRNA combined to the normal stop codon, it will still be released by the termination factors, which stops the full length mRNA translation.
John.
So the biochemical and physiological function of most of the vast multitude of tRNA modifications remains obscure. In E. coli and yeast, it has been typically extremely difficult to find any kind of phenotype associated with eliminating the enzymes performing the modifications. I'm inferring from what you wrote there that you have tested a limited number of modifications, and I'm guessing some have a bigger effect than others. Is that something you've looked into? And then also, have you directly looked at the half-life of the tRNA as opposed to the kinetics of the recovery process?
Yep. Thank you for your question. So the first question is about the chemical modifications. Right. So because of the patent issue, we didn't really disclose all the modification details here. But I can tell you, for example, so first, the rationale of choosing those modifications is because we did it on a glycine-based tRNA. So we choose those modifications that is commonly found in eukaryotic glycine tRNA first, such as M1 gene.
All of them?
Yeah. Huh?
All of them?
No, not all of them. But we do choose the commonly persist ones. Of course, we didn't explore all of them, which is probably impossible, and also some synthesis doesn't allow it. Also, this is not enzymely introduced modification, but we solely synthesize those suppressor tRNAs. So we do see some significant influence on some. For example, one of them, which show almost no restorability, that is pseudouridine modification on the position of 34, which is the anticodon region. There are some previous studies showing that having pseudouridine at the anticodon region could enhance the codon pair. But it doesn't seem to be the case in our case, and also some other modification. But due to the patent issue, I cannot disclose the full detail. But this work is under revision. Hopefully, the paper will be out soon. And in terms of the tRNA half-life itself, so the effectiveness can prolong more than 20 days. We are performing tRNA sequencing right now to see how long this the half-life or the stability of tRNA structure itself. We do see that there's more than 1 month stability, whereas the unmodified sequence the half-life is I think around 14 days.
Next question.
I'm really intrigued by the additive effect of the combination of therapeutics for Trikafta and the tRNA. And I was wondering if you had a comment on how you think that is working.
Yeah, definitely. So the fact that we do see a trend of improvement by the suppressor tRNA treatment
I mean, it's a
itself.
large effect.
Yeah,
if you
it's
look
a
at the
large
graph,
effect.
right?
So we do think that the small improvement that we see from the single treatment comes from the suppressor tRNA raised to the PDC. So there's a higher level of CFTR, full-length CFTR protein expression. But when combining with Trikafta, which has the modulator that help with the CFTR protein folding and processing and localization to the membrane, I think these are playing a more important role in the patient organoids, where we see a more substantial improvement. That being said, a lot of people are comparing suppressor tRNA therapy to directly deliver a CFTR mRNA construct to restore the CFTR functionality, which do not see a lot of success in that case. And we suspect that could due to the fact that those exogenous mRNA that we deliver, those CFTR might not be functionally or like properly folded or processed that localized to CF membrane. And that's where the Trikafta might help in this case.
Eric, yeah, you still have time.
There's a lovely presentation.
Thank you.
I wondered if you could elaborate a little bit on your studies with the cystic fibrosis mice, the R553X mouse. Is that just is that a knock-in mouse? And your readout was just mRNA level, is that right? So you don't know anything about efficiency that the LNP is delivered by, those kinds of things? Or?
Yeah, so this mouse model is a exon 12 replacement. So basically, this is from Case Western University from Dr. Craig Hodges. He's very generous for sending us breeding pairs to start our own colonies. So basically, the mouse CFTR, the exon 12 was replaced with a human exon 12 that contains the R553X mutation in it, which will result in a lower expression of the CFTR mRNA, of course. So we did RT-qPCR to see the mRNA expression level in the lung. We also performed Western blot. However, probably due to the antibody issue right now, we didn't find a really good antibody that could detect the CFTR protein level in those mice lungs. But the qPCR was enough to tell the CFTR mRNA expression, which probably due to the fact that the suppressor tRNA can reach through the PDC of the CFTR mRNA, which prevent a phenomenon called nonsense mutation decay, so that we have a higher mRNA level for translation.
Right. So mostly an mRNA.
Yeah.
Thank you.
Yeah.
So thank you very much. We thank Charles again, and we move to the next presentation.
Yeah.
Okay. So next, I'd like to welcome Soo-Hee Chang.
Hello. Thank you so much for the opportunity to speak today. I'm very excited to share my recent results on how nascent peptides, nascent protein regulates the ribosome and in turn regulates the mRNA stability. My disclaimer slide. So to begin with, why ribosome? And we all know that CFTR is notoriously difficult protein to fold. And among them, the misfolding variant F508del CFTR mRNA level is lower than the wild type. Increasing the mRNA level of these misfolding variants would benefit all of the pre-existing modulator therapies by offering better base, more substrates to work on. And what regulates the mRNA? Actually, the ribosome movement, to be specific, regulates both the mRNA abundance and the protein folding. Therefore, I would say understanding ribosome regulation understanding ribosome regulation would help us to find the next strategies to tackle all the other CFTR misfolding variants. As I said before, ribosome is truly a master regulator of mRNA stability because any abnormal ribosome transit is detected by this mRNA decay pathways I listed here. A lot of stuff in translation can cause the abnormal ribosome movement, such as the previous speaker said, the premature termination codon or the codon usage, tRNA or amino acid availability. Anything happens to cause the ribosome to move weird, and that causes the mRNA to just go trash. However, the role of nascent polypeptide, the key component of translation, the ultimate outcome of the translation actually, that's bound to the ribosome whole time in regulating ribosome has not been widely explored. And today I'm going to talk about how this regulates the ribosome. So to explore this question, I turned to yeast, my favorite organism, the robust genetic system that's easy to manipulate. And I made this artificially misfolded protein by shuffling the yeast history gene. And AlphaFold predicts that this shuffled reporter will make a really misfolded shitty protein at best, by any chance. Does this make mRNA decay faster? The answer is yes. This is northern blot showing that how this mRNA goes away. And you can see the shuffled misfolding protein making reporter mRNA is decaying faster than its endogenous copy that's well folded. And as a result, the steady state abundance is lower now. So yes, the protein product misfolding can affect the mRNA stability. How does that happen? Because there's a physical barrier for the nascent peptide to overcome to regulate ribosome, because nascent peptide is coming from the ribosomal exit on top of the ribosomal large subunit. And the ribosome speed is actually decided how fast the decoding event happens. And that takes place in the A site, in the small subunit. So in order for the nascent peptide to actually affect the ribosome transit, it has to overcome the barrier, this physical distance. And there's a perfect protein for that. So this ribosome-bound chaperone, Zootin or ZIL1, this protein interacts directly with the nascent peptide in one end, in the large subunit here in the ribosome exit, and the other end of it interacts with the ribosomal RNA helix 44 that comes from the A site here. So this protein has a very good chance of mediating the communication between these two sites. Does it? It's also yes. So deleting ZIL1 stabilized the shuffled reporter mRNA. You can see the decay rate is now back to slow, like the endogenous well-folded copy. And the steady state abundance of this mRNA, you can see that it's now back up compared to the wild type in zone deletion cells. And we want to see if it's in general protein misfolding that rules this, or the co-translational misfolding. So we use the GFP protein because GFP matures only post-translationally. It stays unfolded during the translation. And the GFP mRNA is actually stabilized by the ZIL1 deletion, which means that it is indeed the co-translational protein misfolding that ZIL1-mediated mRNA decay is sensing. So cool, we wanted to see if this can apply to the entire transcriptome. So this time we treated cells with the AZC. It's a protein analog that induces the protein misfolding. And you can see that this protein misfolding triggers a bunch of transcriptomic response. So in addition to this heat shock response, it's a stress response to misfolded protein. There's a whole wide range of transcriptomic change in response to that in the wild type cells. It was shocking because in ZOO1 deletion cells, that does not happen. It's completely gone because you can see like the protein misfolding is happening just fine. You can see that stress response is induced here, labeled black. So protein misfolding is happening, but all the downstream transcriptomic response is not happening in the absence of ZOO1, as if cell doesn't know what's going on out there. We did take a closer look into what these genes were. So we filtered these genes by downregulated by protein misfolding, stabilized back up in ZOO1 deletion. These genes actually have higher codon optimality, which is supposed to stabilize these genes. However, the opposite, they actually have shorter half-life. So these genes are these mRNAs are processed by something else than the codon optimality, likely the protein folding. So we wanted to take a closer look at how these shuffled mRNAs, misfolded protein encoding mRNAs are processed. So we used this, we went back to the reporter, and now we started seeing this decay intermediates. So only in the shuffled reporter cassettes, we started seeing this temporary, smaller decay intermediates that disappears over time up in the synchronized transcription of this shuffled reporter. And this is northern blotting, by the way. And then this probe is in the 3'. So this is a smaller portion of this original decay and the original transcripts. And it's probably processing from the 5' and getting blocked somewhere in the middle, blocked by something, likely the ribosome, because when I separate these by the sucrose density gradient, the decay intermediates are found in the ribosome bound fractions. So we can say that ribosome is bound in the middle of this transcripts and blocking the decay from the 5' end. Indeed, deleting the 5' to 3' exonuclease XRN1 and NACFC that detects the slowed ribosome and recruits the decay machinery such as XRN1, deleting both of them gets rid of the decay intermediates. So we could conclude that, okay, this is a product of NACFC sensing the stalled ribosome and recruiting XRN1, and it's chewing from the 5' end and getting blocked by the ribosome. Deleting ZOO1 also decreases the 3' decay intermediates in both the shuffle reporter and the GFP reporter. So it means that the cell needs a ZOO1 to stall the ribosome that will actually process this mRNA through the XR-mediated mRNA decay. And this was dependent on ZOO1's ribosome binding because ZOO1 mutants that cannot bind the ribosome couldn't rescue or bring the decay intermediates back as the wild-type ZOO1 did. So we can conclude that ZOO1 bound to the ribosome is required for this ribosome to stall in response to protein misfolding. To take a broader look into the entire translatum, we adopted this technique called ribosome profiling. Nothing fancy. We're just looking at the mRNA happily translated by a bunch of ribosomes. We treat them with RNAs, and only the mRNA fragments that's bound by the ribosome and protected will be left after that. And we sequence them. So this way, we will have a snapshot of where the ribosomes were at the moment of cell harvest. And the ribosome profiling experiment gave me two interesting things. So here, the y-axis is it's normalized to the unstressed condition. So we induced the global protein misfolding by heat shock and oxidative stress, two different stress conditions. And we normalize it into the unstressed condition. And in the x-axis, you're looking at the beginning of the open reading frame, so from the start codon. The first thing we were excited about was in the wild-type cells, the ribosomes are piling up under the protein misfolding condition. They're all piling up in the beginning of the open reading frame in response to heat shock or oxidative stress. This is completely gone in the ZOO1 deletion cells, as if the ribosomes just don't know what's going on out there because there's nothing to let them know. Like there's no ZOO1 to let them know, like the protein is really badly misfolding on the top of their head. So we took a look at what kind of genes are these. So not all the genes have this ribosome stall. We filter these genes that contains this ribosome pause in response to protein misfolding, like that. And this is connected to the mRNA abundance as well because these genes with the ribosome stall now are downregulated by the protein misfolding. And this does not happen in the ZOO1 deletion cells. They're completely just gone because there's no ribosome stall and there's no mRNA decay. So this ribosome stall is indeed tied to the mRNA decay or stability as well. We also measured the direct ribosome movements using the S35 labeling. So this time, the cells are labeled with radioactive S35 methionine cysteine. Half of them I will collect for the total S35 incorporation into the protein population. Half of them will go through the sucrose cushion, and I will only collect, so this pellets up the ribosome, and I will only collect the ribosome-free, completely released protein population. And you can see that here, the relative total incorporation of S35 methionine is not affected by either the protein misfolding or the presence or absence of ZUO1. So they're all just fine. They're incorporating it as no problem in total. However, it's more interesting in the release fraction because now you can see that this one, it's the wild type in the protein misfolding condition. So the proteins are getting released so much slower than unstressed condition because now the ribosomes are slowing down under the protein misfolding, and this does not happen in the ZUO1 deletion cells because ribosomes have no means to know that they are actually making the misfolded protein and they need to slow down. So we could conclude that ZUO1 is indeed required for the ribosome to sense the protein misfolding and slow down in response to that. So how does ZUO1 do this? So I have an interesting hypothesis for this. This is a cryo-EM study from 2 years ago. And they purified the ZUO1 bound ribosomes from thermophile yeast. And they could purify 2 different conformations of ZUO1. The major differences here include, so ZUO1 interacts with different ribosomal protein in 2 different conformations. In one conformation, it interacts with 31. The other interacts with 22. While doing this, while switching between these 2 forms, it actually tweaks the helix 44 that's connected to the A sites. So it moves a little different angle here, opening the possibility maybe like switching these forms to different ribosome conformations have different A site function. That could affect the ribosome speed. So I just want to test, are they really different in terms of regulating this mRNA? So the answer is also yes, because when I deleted RPL31 that interacts with ZUO1 in only one conformation, that stabilizes the shuffled mRNA that makes the misfolded protein, not 22. So I would say the 31 bound form could be more important in regulating this mRNA making the misfolded protein, not the other. So I was really excited to test this in the CFTR because CFTR is a perfect model of misfolded protein, and it has also already well-established misfolded variants. So I treated the siRNA against ZUO1 in the 16-HBE cells. That stabilizes or increases the level of the F508del CFTR mRNA, not the wild type. So you can see here, it's increasing only the F508del. This is more exciting because it actually synergizes with the modulator in the primary human nasal epithelial cell. So these cells were from the homozygous F508del patient. And you can see here, this is the relative CFTR function. I normalize it to the scramble plus modulator. And the x-axis, you can see that DMSO control, modulator single, and the scramble control plus modulator, and the ZUO1 knockdown plus modulator. And you can see that the modulator and the scramble control don't really have any differences. However, when you knock down ZUO1, it actually enhances the response of the F508del CFTR to the modulator. It could be either the mRNA increase or it probably affects the ribosome movements on this transcripts. I still need to test that. This is really preliminary, but I'm so excited to test this on the other misfolding variants of CFTR, as well as I need to look at the ribosome speed under the ZUO1 knockdown on the CFTR mRNA in different variants. So hopefully today I could convince you how this works in the nascent chain misfolding condition. So in normal translation, ZUO1 doesn't have much to do. It just binds there and then just goes peacefully. But in the nascent chain misfolding condition, this is blocking the ribosome tunnel or changing the ribosome conformation that's getting sensed by the ZUO1 that causes ZUO1 to switch the conformation between, and then that triggers the ribosome to stall. And this is just read by the pre-existing mRNA decay pathways involving XR1 and NOT5. And this could be helpful for dealing with any other CFTR variants that involves protein misfolding or ribosome speed dysregulation and needs an mRNA level increase to help. So thanks for your attention today. And this is my lab, and it's in collaboration with Kristen Kuma's lab in University of Michigan, Ann Arbor, and Niraj Sharma's lab in Johns Hopkins University. And thank you for the CF Foundation for funding and NIH. Oh, yep, I'm done.
Thank you very much. So the talk is open to questions. Margarida?
Hi, Margarida Amaral from Lisbon University, Portugal. So I think this is an excellent presentation, so congratulations.
Thank you.
Maybe you said this, but I was wondering whether the abundance, so the levels of F508del mRNA also increase when you knock down ZUO1. Because for years ago, we found out in native cells from people, not people with CF, but carriers of CF, there was always and consistently an imbalance of wild-type CFTR versus F508del. And this could well be the reason, because we found no difference in promoters or, 5' untranslated regions.
Yeah, exactly. So this one, so it's in the genetically edited cells, but it did rescue the CFTR mRNA in the F508del.
Right.
Yeah, I haven't measured the ribosome speed on this though, so I don't have an evidence that that's how it rescues it. However, this could be an answer. And and I was reading why F508del is lower than the wild type. It's like 80% of the wild type. Right?
Yes, about that. Yes.
Yeah, so this could be it.
Very good explanation. Yeah. Yeah. So I was wondering, the relative expression was relative to F508 itself, but does it also equal the wild type?
Oh no, this is a normalized scramble because I don't have a heterozygous. I don't have the blessing of the patient population cells. So yeah, this is all just homozygous F508del. I think it's just like one allele is silenced in this cell.
Yeah, it's one allele.
Yeah.
Thank you very much.
Gabby?
Yeah.
So hey, that's really nice work.
Thank you.
Has ZUO1 been split and each of the binding domains been used independently to look at this linkage concept and look at stoichiometry with how much you put in of one versus the other, right?
Yeah.
You pose it as a single molecule that's linking the two domains. But obviously there's binding that's going on, probably with different efficiencies to those two sites.
Yeah.
Has somebody worked this out yet, or have you considered think of that? Because I'm thinking of ways to engineer this to be maybe more efficient or less efficient way to get at it pharmacologically.
Yeah, thank you. So I am actually making a bunch of mutants that have different affinities or like even different ability to tweak between two conformations. And ultimately, it'll be really nice to have some kind of small molecule screen to test how to, because deleting ZUO1 itself, an unstressed condition really doesn't do much. It's very, very mild phenotype, actually. Yeah. So if we can find the fine-tuning way of modulating ZUO1 like that, that would be really nice. It's not going to harm the normal physiology at all. It'll just affect the misfolded protein. So yeah, on their way. Thank you.
One final question.
Okay, quick. So does the ZUO1 has a homology in human as well, like compensational mechanism there? And the second question is, why is 85% for delta alpha 8? Is it that system is not efficient to detect the delta alpha mRNA, or not detect the delta alpha CFTR protein, so that not able to degrade everything?
Yeah.
Why is 85%?
So yes, the first question, yes, it is well conserved in humans. There is some variability in the end of the protein, but the basic structure of binding to the ribosome is not changed at all between yeast and human. And the second question, we don't know. So I'm not showing it here, but I made a bunch of different shuffled reporters, and they respond all differently. So there's some domain or some kind of characteristics of the protein misfolding that ZUO1 is sensing. And we did a riboseq, and then we tried to tackle that in a bunch of basic amino acid and tryptophan. I don't know, that's another remaining question. I need to figure out, what part of the translation or to protein domain that ZUO1 is very sensitive to. And I'm hoping I could get the selective riboseq done for that purpose, but TBD. Thank you.
So we thank Sui again and move to the next speaker.
All right. And next, I'd like to welcome Jonica Brown.
Good morning, everyone. Thank you to the chairs for this opportunity, and I'm so excited to talk to you today about my project titled The Role of Cholesterol on CFTR Single-Channel Characteristics and Potential Cholesterol Binding Domains. I have no relationships to disclose related to this presentation. We all know cystic fibrosis arises from dysfunction in the chloride ion channel CFTR. But I'm here to remind you that CFTR is a membrane protein. Thus, its synthesis, gating, conductance, and pharmacology might be impacted by its surrounding lipid environment. Previous work in the Chen lab show a cryo-EM structure, which I'm showing on the left, of CFTR with cholesterol labeled in blue and other lipids labeled in pink, tightly interacting with the transmembrane domains of CFTR. We know from literature that cholesterol does regulate other ion channels through one or a combination of these methods shown here. Membrane mechanics, a direct interaction, membrane localization, or signaling cascades. Reports both in and out of our lab have shown that CFTR is sensitive to membrane lipids and lipid-mediated signaling by mechanisms that remain unclear. Thus, the hypothesis for this project is that cholesterol directly interacts with CFTR at multiple as-of-yet unidentified sites, and its abundance is modulating both the function and structural dynamics of CFTR. To address this hypothesis, I'm utilizing electrophysiology techniques to directly measure CFTR current. That's what makes this project unique. Rather than measuring a byproduct or an indicator of CFTR activity, I can measure a current on a single channel level, such as in a method called planar lipid bilayer reconstitution, where I take a single protein and I embed it into a synthetic lipid bilayer of different lipid compositions, and I can ask, how does CFTR single channel characteristics, such as how long it's open for or how many ions are coming out at once, change between different lipid environments? At the same time, I can use a whole cell approach where I'm expressing different versions of CFTR onto the membrane of frog eggs, and I can measure the current from millions of channels acting together. With this project, I'll be determining where cholesterol is binding based on how different mammalian orthologs of CFTR respond to cholesterol abundance. First, let's dive into this PLB method. I'll be using two phospholipids that are commonly found within our cell membranes, phosphatidylethanolamine and phosphatidylserine, which for the remainder of this talk, I will be referring to as PE and PS. And again, we're asking how different ratios of PE to PS with and without cholesterol impact CFTR single-channel characteristics. First, I create a synthetic lipid bilayer of different ratios of PE to PS. And then I add membrane-derived vesicles stably transfected with wild-type human CFTR into the mix, and these vesicles then fuse with my bilayer. Here I'm showing the three major components of CFTR, the R domain, nucleotide binding domains, and transmembrane domains. And we know for activation, the R domain must be phosphorylated via pKa. The second thing that must happen is two ATP molecules have to bind to the nucleotide binding domains. This channel is now active. To aid in the movement of ions, I add both a chemical and electrical gradient. Then I have a reference and a recording electrode that can measure the change in current once CFTR is active, and it will report it back to me. On the following slides, I will be showing traces of single-channel current, but before we get into that, I'd like to give a brief overview of how to interpret them. Here I'm showing the closed and open conformation state of CFTR. I'll then show you a trace of current on the y-axis versus time on the x-axis. When CFTR is inactive, the current will be positive, When it is active and open, we'll see a more negative current, and then it'll close back. And the two characteristics that I will be analyzing are conductance, or the amount of ions that are coming through the channel once open, and open dwell time, or gating, how long the channel is staying open for. Now that we know that, let's ask our first question. How does the lipid environment impact wild-type human CFTR single-channel characteristics? Here, this is a trace showing current versus time, where current on the y-axis, each tick is 0.5 picoamps, and on the x-axis, each tick is 5 seconds. This is a microsome containing wild-type human CFTR in a bilayer composition of 8 PE to 2 PS. And we see that the single-channel unitary conductance is about minus 0.6 picoamps, while the average open dwell time is around 200 milliseconds. We can then compare this to a different ratio. Now we're observing 7 PE to 3 PS, where the single-channel unitary conductance is around 0.3 picoamps, and now the average open dwell time is around 600 milliseconds. These data were repeated to reach statistical significance for both conductance and gating or open dwell time, concluding that the lipid environment composition does play a role on the conductance and gating of wild-type human CFTR. But now let's ask, how does the addition of cholesterol now impact these characteristics? The goal of this experiment was to take this composition, 7 PE to 3 PS, with the goal of seeing if we can increase the conductance back to 0.6 that we previously observed. Rather than an increase in conductance, we're actually observing a change in channel gating, where the average open dwell time is now around 70 milliseconds, with the caveat that this bilayer may contain more than one channel. Even when we lower the cholesterol content, we are continuously seeing this change in channel gating. These very preliminary data are suggesting that cholesterol abundance impacts the gating of wild-type human CFTR. Now that we have more understanding of single-channel characteristics, let's switch to the whole cell experiment, where we'll be using a combination of 2 electrode voltage clamping and comparative pharmacology with the goal of determining where cholesterol is binding. You may ask, what is comparative pharmacology? This is a method where we take different systems and we expose them to the same environment and we see how they differ in response. We can then search where the differences between the systems are for a potential mechanism. For example, we'll have human CFTR, which will have a response to a normal cholesterol environment and a response to a partially depleted cholesterol environment. We can then take pig CFTR, which has a 92% sequence homology to human, and we would expect to see similar responses. Then if we compare mouse CFTR, which has a 78% sequence homology to human, we may see a change in partially depleted cholesterol environment. We can then search the sequences for sites that are biochemically similar between human and pig and different in mouse. To set up these experiments, I have RNA that encodes for human CFTR and RNA that encodes for mouse CFTR. I then express those separately into xenopus oocytes or frog eggs. And then I separate into 2 groups, a partially depleted cholesterol environment that is obtained by exposing these frog eggs to a cholesterol-depleting agent called methyl beta-cyclodextrin, or the normal cholesterol environment, which receives no treatment. And then I perform 2 electrode voltage clamping. Again, a brief overview of this experiment. We have 1 electrode, which is clamping or holding the membrane potential to a fixed value. In this case, we choose minus 60 millivolts. And once there's a change in membrane potential in response to channel activity in the membrane, the second electrode will measure this change and report it back to us. Again, I'll show you a trace of current versus time, where a more positive current is indicative of less ions, less channel activity. A more negative current indicates more ions, more channel activity. The first question we asked was, how does cholesterol abundance modify human CFTR behavior? This is a trace of current versus time in a normal cholesterol environment. We activate CFTR with forskolin, and then we use CFTR potentiator VX770, which we know increases the amount of ions that flow through the channel once it's active. And this is exactly what we're seeing here. Now let's compare this to a partially depleted cholesterol environment where the only difference is the exposure to M-beta-CD. Again, we see a response to forskolin, but now we're seeing an even greater response to VX770. This is indicating that methyl beta cyclodextrin treatment leads to modification of wild-type human CFTR behavior, specifically sensitivity to potentiation by VX770. To quantify these results, we calculate percent potentiation, which is the amount of the total current that was able to be potentiated by VX770. We see here that these results were repeated with two different forskolin concentrations because we know VX770 is sensitive to phosphorylation. Ultimately, again, treatment with M-beta-CD does lead to modification of wild-type human CFTR behavior. To continue with comparative pharmacology, we then need to ask, how does cholesterol abundance modify mouse CFTR behavior? Again, this is a normal cholesterol environment, and we see both a response to forskolin and VX770. And similar to human, with the partially depleted cholesterol environment, we're observing an even greater response to VX770, indicating that M-beta-CD treatment also leads to modification of wild-type mouse CFTR behavior. These preliminary data suggest that mouse CFTR has a greater sensitivity to cholesterol abundance as compared to human CFTR. With that in mind, we can then go back to the comparative pharmacology experiment, and rather than searching random sites, we will be strategically searching for extensively studied and well-defined cholesterol binding sites termed cholesterol recognition amino acid consensus motifs, or CRAC, and its mirror image CARC. A CRAC site is defined by either a leucine or a valine, separated by 1 to 5 additional amino acids, and then either a tyrosine, phenylalanine, or a tryptophan, another 1 to 5 amino acids, and then it'll end in either a lysine or an arginine. And a CARC site is just its mirror image. So I used the bioinformatics approach to search human CFTR sequence for these sites, and in total I found over 50 combined CRAC and CARC sites. However, since we're interested in a direct interaction between cholesterol and CFTR, we narrowed these sites down to locations that are most likely where CFTR and cholesterol are interacting within the plasma membrane. Thus, based on location, 4 CRAC and 9 CARC sites were found within the human CFTR amino acid sequence. Future directions of this project include to continue exploring the impact of cholesterol at different concentrations on wild-type human CFTR single-channel characteristics, to continue comparative pharmacology experiments and include various mammalian orthologs, as well as clinically relevant variants F508 delta and G551D, and additional compounds to test CFTR function. I would also like to utilize molecular dynamics to visualize the bidirectional relationship between CFTR and its lipid environment. And finally, to confirm the functional importance of these sites via site-directed mutagenesis. Ultimately, showing that CFTR is fine-tuned post-translationally by its lipid environment opens up new areas for therapeutics, where we can modify cholesterol content through diet, pharmacology, or lipid-targeted therapies. We can improve the function of CFTR and enhance current therapies. I would like to thank my lovely and supportive lab for everything that they do, and my friends and collaborators at Emory University and CFTR, and Children's Healthcare of Atlanta, and of course my funding sources. Thank you.
Thank you very much, Janika. So the talk is open to questions.
Really interesting. When you're adding the cholesterol in the first experiments, again, if I got this correctly, you're trying to measure a single channel, a CFTR channel. The frequency of opening and closing is much higher.
Correct.
Do you know if the total flux in ionic change, right, is equivalent to the I guess the other condition in which you're not adding in the cholesterol?
Right. That is something that I'm going to quantify later on. To measure total current, you need three things. You need to know the how many channels are present, the unitary conductance, and you need the open probability. But to get the open probability, you need to be able to calculate how long the open dwell time. To get the dwell time, you need longer recordings. So these recordings are about 5 minutes, but I would need to get at least like 30 minutes to get a proper.
Okay.
Yeah, proper.
Okay. Thank you. Yeah.
Okay.
That was a really beautiful talk.
Thank you so much.
I'm curious whether the crack and carc sites you identified by sequences, how they might overlap with the position of the cholesterol in various cryostructures of CFTR. And secondly, I'm just curious if you looked at the effect of cholesterol under different conditions, different ratios of P to PS, whether or not there were changes.
Okay. For your first question, does cholesterol interact with the carc sites? It depends on which PDB file or protein data bank file deposited that you're looking at. There are some sites located within transmembrane domains 4, 5, and 8 that do interact with cholesterol. And then in some PDB files, they do not. So that's something that we'll be looking into. And then your second question is, I'm so sorry, I forgot. I don't want to make you get up again. It was, how does the lipid environment change with, sorry. No. Yeah, I can hear you.
And the ratios, the amounts of lipids.
So different conditions had very different effects
Mm-hmm.
on the gating conductance of CFTR, whether or not that plays some role in the effect you saw of cholesterol, whether or not it was always kind of the same.
Right. Thank you for repeating your question. We decided just to start with the 7 PE to PS because we were hoping to increase the conductance back to the 0.6, but we can try 8 to 2. It will get difficult with the lipid ratios because the bilayers will start getting really hard to form. So we do want to stick to maybe 2 or 3 different ratios, but that's an excellent point. Thank you.
So, in a somehow related question here, you tested mainly cholesterol depletion. Did you assess increasing the amount of cholesterol?
So the really cool thing about this cholesterol depleting agent, methyl beta cyclodextrin, is that you can preload it with cholesterol or different analogs. So we do have plans to deplete and replete, but we can also just add cholesterol back in and see if there's an increase with that.
And a question also related to Greg's question. Are there any known CF causing variants in the CARC and crack domain?
They are not, interestingly,
Okay.
which I think is really interesting. I searched for F508 and G551D, and they are not in any of my crack and carcs. So that's actually suggesting that cholesterol, even if we're seeing an impact with wild-type CFTR, we might actually see an impact with the variants as well.
Thank you. So if there are no further questions, we thank Jonica again, and we move to the next.
Thank you.
All right. And next, I'd like to welcome Norman Dallaire.
Okay. Can everybody hear me okay? Great. My name is Norm Allaire. I am a scientist at the Cystic Fibrosis Therapeutics Lab in Lexington, Massachusetts. I want to thank the co-chairs for inviting me here to speak today about our work on assessment of codon optimized CFTR sequences in human bronchial epithelial cells. I have nothing to disclose. To start us, codon optimizations have been widely used in protein biologics or therapeutics production to increase bioreactor yields. Codon optimization has been applied to gene replacement strategies and gene therapy strategies to offset known issues with gene delivery. In fact, many CFTR gene replacement or gene therapy programs utilize codon optimization and/or overexpression. There's been some literature that suggests that codon optimization and/or overexpression may lead to basolateral localization and inhibition of vectorial ion transport. To build on this information and to extend it, we sought to look into the effects of codon optimization on CFTR processing and subcellular localization. So this is a schematic of our experimental design. We took lentiviral constructs, and they were engineered to encode either a codon optimized sequence or native sequences. These are all driven off of the CMV173 promoter. They were transduced into G542X R553X cells in an undifferentiated state. They were then blastocyted and selected for 5 days and then seeded onto filter plates. After 4 to 5 weeks at ALI, the differentiated and selected cultures were then harvested and subjected to downstream analysis. Excuse me. In the table, I'm showing you the sequences that were interrogated. In the royal blue bar, we're going to show you our CFTR full-length sequences. You're going to see this throughout the presentation. We have native sequence. We have HCI, or high codon adaptation index sequence. This is a sequence where it's modified to represent the codon usage bias of the most highly expressed genes in an organism, in this case human. We also had sequences 1 and 2 were different versions of codon optimization or different algorithms for codon optimization. These were obtained either through patent searches or they were shared with us by third parties. In delta R, in our navy blue, we have delta R sequences or removal of 52 amino acids of the R domain. We also have native and HCI controls, as well as a third version of codon optimization. And lastly, a fourth sequence, which is a native sequence which carries a non-CF causing missense variant. So our control cells are normal non-CF HBE cells. We also included HBE cells with the G542X R553X variant or genotype. These were both non-transduced as well as transduced with the non-CFTR gene, in this case Cas9, to control for the effects of the lentivirus on HBE differentiation. The assays that we used, we measured average transduction rate or copy number. We measured mRNA and protein levels. We looked at electrophysiology, or we measured chloride conductance. And lastly, we looked at CFTR localization and imaging, and we measured cell type by immunofluorescence and confocal microscopy. So on the left-hand side, in the gray and the black bars, I'm showing you the copy number of endogenous CFTR in diploid cells, which is 2. And the royal blue and the navy blue bars, I'm showing you the endogenous transgene copy number. And what you can see is that in the full-length and delta R sequences, our transgenes are roughly between 1.5 and 5 copies per cell. So we then use this data to normalize our mRNA data, and you're going to see this throughout the presentation. We're going to normalize all our downstream data by the copy number, and then we referenced it to a single copy of endogenous CFTR. And so all the data you're looking at is on per copy basis. And so if you look at the native full length and the native delta R and our sequence 4, which is a native sequence carrying a non-CF missense mutation, they're about 5 to 6-fold over normal non-CF cells. And so this reflects the differences between CMV promoter and the native promoter, native sequence versus cDNA, and the CMV promoter. So if we look at HCI sequences relative to the native sequence, what we see is a large stabilization of the mRNA on the orders of 20 to 30-fold. And then if we look at our sequences 1, 2, and 3, these are 3 different versions of codon optimization again. What we find is intermediate levels of stabilization. So from this, what we're concluding is that we're getting a large stabilization of our mRNA, and the stabilization is different depending on what type of codon optimization is used. Next, we can look at increases in band C, or mature CFTR, with codon optimization. And what I'm showing you here is if you look at all the codon-optimized sequences, you're going to see a large increase in band C relative to native controls. However, what you'll also notice is that we have a large increase of proteolytic intermediates, or degradation products, with all our codon-optimized constructs. The next, if we look at the absolute increase or absolute difference in protein expression, again, this is all normalized by copy number and in reference to a single endogenous copy. What we see is with our native, we see between 1 and 5-fold increase over our non-CF normal. And then we see a very large increase with codon optimization on the order of 1 to 2 orders of magnitude. So I'm particularly talking about the HChi sequences at this point. And so if you back out the 20 to 30-fold change that we had at the mRNA level, what we're talking is about a 3 to 4-fold change at the protein level. So a disproportionate effect on mRNA stability versus protein for a total change in protein of 1 to 2 orders of magnitude. So for very large changes in magnitude. And as we saw with the other data, sequences 1, 2, and 3 are intermediates of these. So now if we switch over to our electrophysiology traces, on the top we have our non-transduced control as well as our lentivirus transduced. And as you can see, we have no chloride conductance as expected. And then we have our normal non-CF, which has a characteristic trace from normal cells, which is a reduction in ENaC activity with benzamyl, an increase of increase in ENaC with benzamyl, a stimulized CFTR current with forskolin, and then a reduction back down to baseline with our CFTR inhibitors. Now, if you look at our native sequences, and again, our forced sequence, because it's essentially, in my mind, another control, what we see is we see a reduction in ENaC, but it doesn't reach baseline levels. And then we see, when we stimulate with forskolin, we see an increase in current. And then with CFTR inhibitor, we see a reduction below baseline. So what this is suggesting to us is that we have constitutive activation of CFTR, in that it's an improvement in chloride current over our non-transduced control. So we're getting increased chloride current or CFTR-specific chloride current, but it's not necessarily normal physiological CFTR chloride current. If we look at our HChi sequences, both full length or delta R, as well as sequences 1, 2, and 3, which again are 3 different versions of codon optimization, we have the same effect, but it's just more exaggerated. So now if we look at the amounts of chloride transport, again, normalized by copy number, reference to our endogenous CFTR, we have about 6.5 microamps with our non-CF. In our non-transduced and lentivirus transduced, we get no chloride current, which we expect. And then with all cases, we see a detectable chloride current with either overexpression or with codon optimization. And then in the case of HChi sequence 1 and sequence 2, which are full-length sequences, we're approaching wild-type levels or non-CF normal levels of chloride conductance. I'm saying approaching because this reference non-CF sample was low on the scale of chloride conductance relative to other wild types. But what's important to note here is that it's in the range of wild type. It's approaching the range of wild type. The next thing I wanted to point out is if you look at the difference between HChi and native, whether it's full length or delta R, you see about a 2-fold change in chloride conductance. However, in these samples, I've just told you that we have about a 1 to 2 orders of magnitude change in protein. Okay, so there's a disconnect. There's a nonlinear relationship or a disconnect between the protein levels and the chloride conductance, but we're clearly getting an increase in chloride conductance. So now as we move on to the imaging data, on the top I'm showing you our non-transduced control and lentivirus transduced control. And as you can see, as expected, you're not seeing any CFTR-specific staining. If you look at our normal non-CF, you start to see staining of CFTR. And then if we look at our native controls, whether it be full-length or delta R or a sequence 4, we're starting to see an increase in staining of CFTR-positive cells. And then for our HChi and our 3 codon-optimized sequences, we're seeing a further increase in CFTR positivity. One thing that's important to note with all these cultures are grown in Vertex ALI. And we had an underrepresentation of CC10 positive cells in this. And I'm going to talk about that in the next couple of slides, just a point to note. Now, if we look at orthogonal views of these cultures, if we look at our non-transduced and our lentivirus transduced, I'm showing ZO1 staining at the apical membrane, at the apical surface. And as you expect, we aren't going to see any CFTR staining in those cultures. And then as we look at our normal, we see co-localization of ZO1 staining with CFTR, as we'd expect. And then as you look through these cultures, as you look for a CFTR staining, what you'll notice is that in all cases, we're seeing co-localization of green CFTR with our ZO1 apical staining. And so we're not seeing any indication of basolateral localization, which was one of the original thought why we started this experiment to begin with. Okay. So I had told you that we had run our Vertex ALI cultures and we had underrepresentation of CC10 positive cells and also a lower number of ciliated cells. So with this in mind, what we did was we repeated an experiment and then we transitioned into Pneumocult. Pneumocult gives earlier specification and more complete specification of cell types. They also give rise to thicker, taller epithelial layers. And so on the left-hand side, what I'm showing you is our ZO1 staining at the apical surface. And you see a nice staining with green. Okay, so apical localization of CFTR. And then as we transition over to the right side, as we transition over to the right side, what you're seeing is presence of CC10 positive cells in red, presence of ionocytes. In this case, we have an ionocyte with CFTR at the top, as you expect. And again, the story remains the same. We're still seeing apical localization of CFTR. So in summary, the first bullet point is about the effect, the comparison of codon-optimized versus native sequence. And in that case, codon-optimized versus native sequence, we're seeing an 8 to 30-fold increase in mRNA levels, presumably due to enhanced stability. On the protein level, we're seeing a total increase of 1 to 2 orders of magnitude. However, we do see presence of proteolytic intermediates. And then lastly, for that comparison, what we see is about a 2 to 3-fold increase in chloride transport. However, these are nonlinear with mRNA levels as well as protein levels. The next bullet point I want to emphasize is that when we take codon-optimized CFTR and put it into CFTR null cells, CF cells, what we see is we see improved chloride currents. And that hasn't been shown in the previous work. However, even though we're getting improved chloride currents, they don't look like perfectly normal CF currents. So, there's a lot to unpack there. I'm sure you'll have questions. And then lastly, in differentiated HB cells, what we're seeing is apical localization of CFTR, and we don't see any indication of gross mislocalization or basolateral mislocalization. So, I have the privilege of standing up here and talking about these experiments, but the work that was done in this spanned the entire lab. It involved a lot of really bright people, very dedicated, hardworking people. So I just want to acknowledge all their work. I'm not going to go through everybody individually. And then last, I'll leave you with a photo of the CFF lab. This is taken about a month ago during our annual lab tour. And you see a lot of happy, smiling faces because it's a really fun place to do science. And with that, I'd be happy to take any questions.
Thank you very much, Norman. So, yeah, lots of questions coming. Yeah, you may start, yeah?
Yes.
Hello, awesome talk. So if you optimize the codons, it'll definitely change the ribosome speed.
Mm-hmm.
And I remember people did look into the ribosome speed along the CFTR, and some domains are slower than the other.
Yep.
Have you considered optimizing just portion of it?
Yeah, that's a great question. I wish there's a guy that works in my group, Jay Yoon, who's done an awful lot of this work, and he would have a much better answer than I'm going to have. But he did a lot of that, and there are regions that you could clearly codon optimize that improve, like around NBD1, I believe, is one region. And then I didn't mention this, we didn't have time. In our codon optimized samples, the algorithms that were used, there were some regions that were non-optimized and some regions that were. And they behave differently than our maximally codon optimized. So I think you're 100% correct that it's likely that there are regions that are probably more important than others.
Thank you.
Yeah, thank you.
John.
Yeah. Hi, Norm. That was great.
Thanks, John.
When you normalize the number of integrants, do you think that that's fair? So
Yeah, that's a great question.
Have you
Yeah.
tried to go higher with your integrants and see if it's linear?
Yeah.
So are you underestimating or overestimating by dividing by that number?
Yeah. It's a really good question. We've had a lot of these discussions. I'm a technologist, so I like to have everything as head-to-head as possible. We did do experiments where we increased the transduction rate. We saw much the same that we saw in these studies. When I look at the data in an unnormalized state by copy number, the data doesn't make as much sense. It isn't as clean. And maybe that's not a great answer to your question, but, I've seen in the papers I've read, they seem to normalize by copy number and the data seems to make more sense to me. So thank you.
Charles.
Yeah.
Thank you for the great talk. It's very inspiring. So my question is that first, what is the general consideration in terms of for those codon optimization? Is it CAI, of course, is being considered, or MFE, the minimum free energy?
Yeah.
What is general consideration for these?
Yep.
And second question is that is the ORF region and the UTR region has the similar design rationale?
Yeah. Okay. So the first question is about the nature of codon optimization. So these sequences were obtained through patent searches. So we're looking at these very observationally. So they're codon optimized by algorithms, and we don't know all the details about why they chose regions that they chose. What I can tell you is about the HChi sequence, which we know a little bit more about that. And that's codon optimized in a way that they're going to take the most representative codons and the most highly expressed genes. And the idea is that those should be the most abundant and not rare, and that should improve translation rate. So in our HChi sequences, every amino acid is represented by one codon. In all our other codon optimization sequences, that's not the truth. They're represented by multiple codons in different regions in different ways. So, and then I'm sorry, your second question?
Second question is the design rationale for UTR region and the ORF region.
Yeah. So the design rationale, so our original sequence that we're interrogating, we're just looking at the CDS. So we have a WIPRI sequence that's incorporated into the lentivirus. That's a part of the packaging of that virus. We retained a very short 5' UTR that was consistent across all of our constructs. And then the 3' UTR is that WIPRI sequence. So they're consistent, but it's non-native, I would say that.
Thank you.
You're welcome.
Cedric.
Hi, I'm Cedric Cedric Govas from Brussels.
Yep.
So I have a naive question. Maybe I didn't understand. So I have two questions. Naive question is you mentioned increased stability of the RNA. How do you differentiate stability versus translation efficiency?
Yeah.
And the second, can you comment on this discrepancy between the increased in mRNA versus the increase in protein? Because coming back to the speed issue of translation, there's a whole debate whether or not you would get more unfolded protein if you don't get the right speed and right poses.
Yep.
So do you have a way to measure whether you get more unfolded protein at that point?
Yeah, so let me just go back to the first question. You just repeated it. I want to make sure I get it right. The first question was about...
So maybe I don't get the wording.
Stability.
Yeah, you say stability over just more.
Yeah.
And it just...
So when we look at the mRNA, it's a function of... how much, it's a function of the promoter, which these all have the same promoter. So it's how much is made versus how much is degraded. Right? So whether it's a result of, increased speed as previous presenter had suggested, or, depletion of uracil or whatever, we're attributing that to increased stabilization by some mechanism. We don't know exactly all the details of it. And then one more time, your second question?
The second question is the difference between the amount of protein you get versus the amount of mRNA you have.
Yeah.
So whether that's protein gets just unfolded.
Yeah. So again, they both increase, and there's less of a disconnect than there is on the functional level, and there's lots of reasons that we don't fully understand, honestly. They're generally linear, but if you noticed our protein levels, I'm going to go back to that. They have pretty large error bars, right? So, you know, we believe that data, but there are larger error bars, so it's harder for me to kind of comment on, you know, how different they really are versus our mRNA amount. So I would say they generally correlate, but I can't make a really firm statement on like how perfectly they correlate. Thank you.
John?
Does the constitutive activation create ER stress or any degree of toxicity?
So it certainly could. I mean, it seems like it could. You know, we didn't measure that in these studies, but I think that's a reasonable assumption that with 100 times more protein, that could be the case. Yeah.
Colin?
Hey, Colin.
Hey, Norm. Great talk.
Thank you.
So I think a lot about sort of local codon recoding in the context of modulating gene editing strategies. And something that I worry about with that approach is that you can inadvertently introduce splice sites into the sequence. And there are known CF causing alleles that are actually synonymous, but that function pathogenically by introducing splice sites. Do you feel like some of these codon optimization strategies could be changing the kind of splicing and isoform?
I think you're 100% right, and that's something that we're thinking a lot about, and I certainly think along the same lines as you do. And you know, we've done a lot of work on splicing in the lab, and we know that these things happen. And we actually have done, with our super exon approach, we actually have done some codon optimization when we inserted an intron 22, and we did see a lot of splicing liabilities. Now that is confounded by there's some natural splicing liabilities in that region. What we're currently working on is a codon optimized strategy with a super exon where we codon optimize but downstream of the splice site. So if we're going to codon optimize 2 through 27, maybe we'll only codon optimize 3 through 27 to try to preserve that splicing. That's work that Ariel is working on, and we're super interested to see how that's going to work. I'm super excited about that because that gives us potentially the ability to have cell specificity, which I think is super important.
Thanks.
Yeah.
So, I have a final question. Did the IFs that you showed were done under permeabilization conditions? So did you assess if CFTR is really at the membrane, like something like cell surface biotinylation, or?
Yeah. So this is a immunofluorescence question. Is Victoria here? So I'll have to get back to you.
Okay, so I'll get back to you.
That detail. Thanks.
Okay, thank you very much, Norman. So yeah, thank Norman again.
All right. So we will end with the pre-recorded talk from Dr. Deborah Baines. And so naturally, we will not have a Q&A, but that just means we'll end a little bit early.
Thank you for inviting me to this workshop to discuss the development of a tool for in vitro assessment of anion transport in common and rare cell types of the human airway epithelium. I'm so sorry I can't be there in person, but I'm happy to answer any questions out with the meeting. These are my disclosures. So we all know that cystic fibrosis is an autosomal recessive disease caused by mutations within the cystic fibrosis conductance regulator gene, or CFTR gene. CFTR is an epithelial anion channel, and it transports chloride, bicarbonate, but also iodide. Absence of CFTR leads to the pathophysiology of CF disease, and because of this, there are many approaches that are being used, particularly gene therapies and gene editing approaches, to try and correct or replace the CFTR gene, particularly in those for whom the new modulators don't work. But there are cell-specific differences in the expression of CFTR and its function. So really understanding which cell types may be corrected or in which cell types CFTR is being replaced would be very helpful for our progress with gene therapies. So there are cell-specific differences in CFTR expression, and these are just two images from two recent publications, one from Carlos Farina et al. And the other one from Travaglini. And what you can see here is that there are different cell types in the airway epithelium, and they differ in abundance, but also the expression of CFTR differs in abundance in those cell types. On the right-hand side, you can see a slightly more detailed picture showing that in the ionocytes, CFTR is highly expressed, but in other cells, such as mucus secreting cells, basal cells, have a moderate amount of CFTR, and there is still little dispute around whether CFTR is expressed or functional in ciliated cells. So knowing which cells can be corrected for CFTR is important. So the aims of this study were to express a halide-sensitive yellow fluorescent protein, HSYFP, under the transcriptional control of cell-specific gene promoters, and we really wanted to target secretory cells, ciliated cells, and ionocyte cells. And we wanted to express these in a semi-immortalized BMI1 transduced human nasal epithelial cells so that we can use this as a tool. We used 3 different types of immortalized cells. We used non-CF human nasal epithelial cells. We used CF cells with the G542X variant of CFTR, and we used those CF cells that had been base edited to correct the G542X to another form of CFTR, which is functional. Once we had these cells, the next aim was to measure halide-sensitive fluorescent quenching to determine the contribution of CFTR protein to anion influx in these specific cell types. So to do this, we used homology-dependent targeted integration of HSYFP into the safe harbor site of human nasal epithelial cells. So first of all, we identified promoter regions that would produce cell-specific expression of halide-sensitive YFP, and we used previous publications, but also databases, and we used bioinformatic programs to identify 3 promoter regions. So we identified one, which was the MUC5AC promoter for expression in secretory cells. We used the FOXJ1 promoter for expression in ciliated cells, and we used the ASCL3 promoter for expression in ionocytes. And we replaced the promoter in the HSYFP plasmid with these cell-specific promoters. We co-transfected this plasmid alongside a Cas9 PX458 plasmid that integrates the HSYFP into the AAVS1 safe harbor site of the cell. So both of these plasmids contained the guide RNAs to target to the AAVS1 site. But as HSYFP is not expressed until the cells are differentiated into these different cell types, we also used mCherry to identify which cells had potentially taken up these constructs and were transfected. So these 3 constructs were transfected into basal human nasal epithelial cells using lipofectamine. After 48 hours, we identified the mCherry expressing cells and used FACS cell sorting to gain an enriched population of those cells that were expressing mCherry. We then took those cells and we seeded them onto the underside of transwells and grew them at air-liquid interface for 28 days. So we grew them on the underside because we were using a confocal microscope it was an inverted confocal microscope, so it enabled better imaging, and the cells grow quite well in this direction. After 28 days, we then checked the cells for functional cilia and for mucus secretion, and then we started our experiments. So the media that the cells were grown in was exchanged for physiological chloride bicarbonate solution, and we measured baseline fluorescence. This solution was then replaced with 100 millimolar iodide solution, and we also used forskolin to activate CFTR. The cells that we identified were captured as individual cells, and we have taken the mean from fluorescence from those cells, and we then quantified fluorescent quenching by measuring every 2 seconds for a total of 250 seconds. And the data was then analysed using NIH image. We also conducted control experiments where we inhibited CFTR with the CFTR inhibitor 172. We also controlled for endogenous light sensitive fluorescent quenching over the same time course. And we also controlled for vehicles used with the application of drugs. So to ensure that the HSYFP was co-expressed in the cells that we expected it to, we also conducted some immunostaining. So here at the top, we have cells that were HSYFP was under the control of the FOXJ1 promoter. To the left, you can see the cell nuclei stained with DAPI in blue. The HSYFP is shown in yellow. And we immunostained the cilia with alpha tubulin. And then on the fourth image on the right, you can see the merge image. You can see that there are cells with co-localized HSYFP and cilia. Similarly, for the MUC5AC promoter, you can see the MUC5AC there is immunostained with orange, and you can see co-localization with HSYFP. And finally, the ASCL3, you can see in white in the third image across, and this is co-localized with HSYFP cells. And to the far right-hand side is just a video image showing active cilia in these cells, just to show that they were fully differentiated. So to the measurement of iodide influx, so these are in non-CF human nasal epithelial cells, and at the top you can see these are the three constructs: the MUC5AC promoted HSYFP, the FOXJ1 promoted HSYFP, and the ASCL3. So we are assuming, but not that these are secretory cells, ciliated cells, and ionocytes. Below, you can see the fluorescent quenching curves obtained over time. So you can see in the first one to the left-hand side, in the mucus secreting cells or the secretory cells, you can see that there is a decrease in HSYFP fluorescence over time. In the ciliated cells, you can see there is a very slow and shallow decrease in fluorescence. And on the right-hand side, you can see in the ionocytes, there is quite a large decrease in fluorescence, but there is also quite a lot of variation in these cells. To the graph on the right-hand side, we've calculated the maximal rate of iodide influx, and you can see from this that the maximal rate of iodide influx is highest in the ionocytes, followed by the secretory cells, and it's very low in the ciliated cells. And if you look at the total influx, it follows pretty much the same pattern. And this is consistent with current understanding that there are more CFTR channels in ionocytes than in the mucus secreting cells, and very low amounts, if at all, in the ciliated cells. So having identified that we could measure halide fluorescent quenching in these different cell types, and that it was different, we then went on to look at HSYFP quenching in cells that had been gene edited. So these cells were CFG542X variant human nasal epithelial cells, and they had been gene edited, base edited to change the G542X stop codon, in which no CFTR is produced, to a G542R, which produces protein, but it is not as active as the wild type CFTR. Editing was carried out in basal cells, so we assumed that the editing would, in fact, affect all cell types when differentiated. And the editing was 52% of alleles were edited to G542R from G542X. To the left, you can see a graph showing the short circuit current, where there is no CFTR-mediated chloride current in the CF cells, but in the edited cells, you can see that we have restored some current, and to the left is the wild type. Now, the G542R is not as active as wild type, but it does restore some function. The QR code is given there if anyone wants to find out more about that study. So in the cells that we had differentiated, we then looked to see what percentage of cells actually responded to forskolin, and in which we saw fluorescent quenching. In the MUC5AC secretory cells, we saw about 53% of cells started to quench. In ciliated cells, there was about 62% of cells exhibited quenching. And in the ionocytes, we saw, again, about 56% of cells quenching. So this is consistent, really, with the number of alleles, similar to the number of alleles that were corrected. So when we then looked at fluorescent quenching over time, we saw some slight differences and to what we saw in the wild type cultures. So the wild type or the non-CF CFTR, I have put on the graphs here. If you look at the bottom, you can see the original curves in orange, red, and green, and then the curves that we obtained from the corrected cells are shown in red, blue, and yellow. So you can see that we have restored current, or we've restored iodide flux, to the secretory cells, although this is less than we would expect for correcting everything to wild type. We saw a slight change in the quenching curve in the edited ciliated cells, but it still remained very low. And interestingly, we did see restoration of current in the, restoration of flux in the ionocytes, but it seemed to be a lot less than we would have anticipated, given the function of the G542R. So the function of the G542R will be less than wild type. But if you look at the right-hand side, the maximal rate of iodide influx was now greatest in the secretory cells. The ciliated cells still remained as low, the lowest restoration of current and rate of iodide influx. But now the ionocytes don't seem to have restored the maximal rate of iodide influx to the same extent as we would have expected, given the number of channels there. So this is preliminary data. We haven't yet treated these cells with Kaftrio or elexacaftor, tezacaftor, and ivacaftor, which is known to activate the G542R to almost wild type levels. So it will be interesting to see how treatment with the modulators might restore iodide influx in these cells, perhaps back to wild type or not. We still have some additional experiments to do to really understand what is happening here. But in conclusion, or in summary, we suggest that we've generated HETI-edited BMI1 human nasal epithelial cells driving halide-sensitive YFP expression from cell-specific promoters. We observed differences in CFTR-mediated iodide influx between MUC5AC, FOXJ1, and ASCL3 expressing cells. Gene editing of basal CF human nasal epithelial cells G542X2R was observed in differentiated MUC5AC, FOXJ1, and ASCL3 expressing cells. But there seem to be differences in restoration of current between the three cell types that requires further investigation. But we could say that cell-specific expression of HSYFP could be used as a tool for in vitro assessment of CFTR-mediated anion transport in common and rare cell types of the human airway epithelium. And we might expand these cell types to include basal cells and others. So all that's left is to say thank you very much to Scott Randell, who provided the G542X human nasal epithelial cells, to Robert Tarrant, who helped with the analysis of some of this data, and to Emily Woodcock and Nikita Demenchenko from St George's, who helped with the imaging at the imaging resource facilities, and also members of the Cystic Fibrosis Strategic Research Centre, O2O, for their helpful insight and advice. And thank you for listening.
So this brings the session to a closure. Thank you to all the speakers, and thank you all of you for coming and for the exciting questions. Enjoy the rest of the conference.